Oliver Sartor presented SPLASH at ESMO, which is on the right-hand side here, rPFS and OS. You see rPFS hazard ratio 0.71, and note the medians there of 9.5 and 6, which are different than we saw with the lutetium data, and then no OS difference, right? Those overlapping curves. And the discussant, made a point that it was all about dose, right? That's sort of what I took from that discussion, that it was all about dose. And you see sort of the dose calculations on the bottom there, 44 versus 27, 40% difference. So I guess, is it true that dose matters, right? And maybe, Gary, I'll start with you. How do you think about, and we're balancing toxicity, as mentioned, cytopenias. Do we know what dose to give? I mean, we're giving the standard dose, but it seems to me that we're just sort of starting to learn about this dose effect.
Yeah, so I actually was at the Society of Nuclear Medicine Therapeutics conference last week in Washington, and one of the presenters presented data to suggest that if we look at total body burden of disease as opposed to single lesion SUV, that potentially will correlate better with response. It's actually inversely correlated, as might be expected, that the higher total body burden of disease, if we're given a single dose of 200 millicuries or a slightly reduced dose of 160 of the Pluvicto, that's distributed across more lesions and has less total radiation. So it may be that what we needed to be doing is, again, this dosimetry step to try to predict the dosimetry to individual lesions and specific lesions. One of the difficulties with that is that until we get dosimetry-specific guidance on dosing, rather than giving a single dose of 200, that it may not make any difference to do that. But I think that there is data to suggest that the dose is important, and the difference between these is significant, I think.
And I don't know enough about the other agent and how it was developed. I mean, why was it a lower dose? I mean,
this is the intersection of clinical science and regulatory science. This dose was reduced by the agency, basically, out of concern that the patients would get too much radiation, and that was...
Even though it's less than...
Well, that was regulatorily determined, that the dose couldn't be 7.4, had to be given at 6.8 times 4, as opposed to times 6, and given it Q8 weeks rather than Q6 weeks. So I think that this is an ongoing discussion. There was a big meeting this spring with NSNMMI and the FDA about how to approach this issue in the future, and where the line is, where you say, well, I'm going to try to spare the patient risks of side effects from radiation, but then you expose the patient to the risks of poorer tumor control.
Right.
And it's that balance that I think that everyone is now struggling with. And unfortunately, I do think SPLASH was the victim of being one of the first to be in that battle.
Bad timing.
Yeah.
But I think, as we talked about with the bispecific T-cell engagers, we are getting better with subsequent iterations. We're tweaking, and there may be something about how the target is bound by the drug, as well as different particles. Like lutetium PSMA is wonderful, but it's not the end. It's the beginning of this frontier, and we need to continue to develop new doses, new strategies, and new agents that will get more dose to the tumor.
The targeting agent for SPLASH is not PSMA-617.
Right.
So that raised some concerns from a regulatory standpoint.